Hello, thank you so much for having me here to talk about planning reproducible, replicable, robust, and generalizable studies. Really excited to be recording this talk for you. And if you want to follow along with the slides, there's a bit.ly link here. There's a few links in the slides that might be helpful, so that might be useful to get down. So I like to start by just introducing who I am. So I'm Priya. I did my PhD training as a developmental cognitive scientist. So I was studying how infants learn that they're being communicated to. But then since my PhD, I've been working predominantly in meta-science. So I study the topics that I'm going to be talking to you about today, but I'm not going to be presenting any of my own work today. So because I've kind of opted for a more kind of biomedical focus because of the audience. But I study replicability, reproducibility, and generalizability in psychology and other social sciences. So at the moment, I'm looking predominantly on generalizability. So I'll talk a bit about that at the end. But kind of most relevant for this talk is that I'm just a massive open science nerd. I love teaching other people about how and why to adopt open science practices in their own work. So hopefully I can convince you of the benefits of that today. So I'll start by talking about what we should be aiming for and what the kind of differentiation is between these four terms that I'm going to be talking about. And I really like this little table from the Turing Way, which is a great resource for anything to do with open science. I'd recommend checking it out. So we have these kind of four quadrants of good research. So what they are reproducible, research should be reproducible, replicable, robust, and generalizable. And these can be differentiated in whether or not they use the same data or different data than a kind of original reference study, and same or different analysis to a reference study. So if something is reproducible, it means that you should be able to get the same results when you run the same analysis on the same data. Whereas if it's replicable, it should be that you get the same results when you run the same analysis on different data. And kind of robustness and generalizability are kind of the other side of that. So when you have a different analysis but the same data, you're looking at the robustness of the original result. Whereas if you have both different data and different analysis, you can be looking at generalizability. So I'll go into a bit more detail about each of these four as we go through the presentation. So we'll start with replicability, because I think it's kind of important for understanding the context of the others. So I have used definitions throughout this talk from the Fort glossary. That's another resource that I'd really recommend. So it's a glossary of open science, a crowdsourced glossary of open science terms. But they have quite a extensive definition for replicability just because this word has been used very differently across different fields and by different people. So I've kind of crossed a lot of it out because it refers to parts that are actually better defined by the other terms that I'll be talking about today. But replicability in general covers the concepts of direct and conceptual replications. I'll talk a bit about the difference between those later. But some good definitions are a study arriving at the same conclusion after collecting new data. So that would be if something is replicable. And also studies for which any outcome would be considered diagnostic evidence about a claim from prior research. So this is kind of a long-winded way of saying, conducting the same study again, but it kind of needs to be long-winded because of all the caveats of what it means to conduct a study again, which we'll talk about. So what's going on when a study doesn't replicate? Well, there's a few options. So the first option is that the original effect is true, and the replication is kind of isn't. So in this case, if the replication result could be a type 2 error, so a false negative. This could be the case because the replication is underpowered, so it didn't have a large enough sample size to detect the effect. And this can be especially true if the true effect is smaller than in the original study. And the way that this happens is that either through randomness or through kind of things that the original researcher has done, they kind of might have had an inflated effect size estimate in their original study compared to the true effect that is in the world. And so if you then use that original study to do a power analysis and decide how big your sample should be for the replication study, then you might actually be underpowered even though you did perform a power analysis. So that's one reason why you might get a non-replication. Or it could be that the replication is correctly powered, but just didn't detect the effect, just because of kind of the chance and the fact that that's how inferential statistics work, is that some of the time you will get a type 1 or type 2 error. On the flip side of this, it could be that the original effect isn't true, there is no true effect, and the replication is kind of right in that way. So the original result was just a type 1 error, it was a false positive. So this will happen just generally because when everything else is equal, this will just be the alpha rate. So you will just kind of get some of the time that you will have a type 1 error. But this can be made much worse through questionable research practices. So through things like p-hacking, where you kind of do lots of different types of analyses and ways of conducting your study and then only report the one that kind of came out statistically significant, or through harking, which is hypothesizing after the results are known, where you get your results and then kind of come up with a hypothesis afterwards for why you might have got those results, and frame it as if you kind of went into the study thinking that. Things like this can inflate the type 1 error rate, basically make it more likely that you get a false positive. So if the original study had done any of these questionable research practices, either intentionally or not, then it could explain why the replication doesn't find the same effect. But it also doesn't have to be either of these dichotomous things. It can be that it's complicated. So differences between the studies could be responsible for the different results. So differences in the design, in the participants, or the sample used, and the analysis can all be responsible for getting different results in two studies. So with this in mind, for what kind of, how to explain non-replications, what does this kind of look like? Does this even matter? Does this happen very often? What % of findings cannot be replicated? And this has been investigated across lots of different areas. So in psychology, we found that up to 60% of findings cannot be replicated, which was a big kind of shock to the field when it originally was found. In economics, up to 40%. In experimental philosophy, up to 30%. And in cancer biology, which I'm going to be talking about the results from this study quite a bit in this presentation, up to 55%. So I think for a lot of people in the social sciences, this was kind of a big shock, because there's kind of this implicit idea that like the hard sciences are better, would be better with regards to replicability than the social kind of more fluffy sciences, where so many things could, affect the results. So I think just from a kind of like intuitive, anecdotal perspective, this was quite a shock to a lot of social scientists when this result came out. So this result came from the reproducibility project, Cancer Biology. So I'll be talking about a couple of papers that came out of this project. So RPCB aimed to assess preclinical cancer research replicability. So it repeated experiments from high impact papers in this area. 50 experiments from 23 papers were repeated. So within this, there were 158 effects. Most of these were positive, so like significant in a particular direction, but some were null. And they used 7 different methods to assess replicability, because they weren't all suitable for different effects in the sample. So they kind of wanted to have a bit of a range. Okay, so I'm just going to explain this graph because there's a lot going on. So this is a graph comparing the original effect size within the original studies and the replication effect size. So it's important first to differentiate between the blue and the pink circles. So in the blue circles, this was where in the replication there was a significant result in the same direction as the original. There was a significant result in the same direction as the original study, whereas the pink dots is where they found a null result in the replication. So the first kind of, oh, and these, the left and right are the same. It's just that the right is a zoomed in version. So the right is this, what's contained within this rectangle, just zoomed in so that you can see it a bit better. So the first thing that we notice is that there are a lot of pink dots. So there are a lot of failed replications. And the other thing to note is that even of the ones that did replicate kind of in terms of a statistically significant result in the same direction as the original, so the blue dots, these are very much clustered below the diagonal line. And so what this means is that effect sizes were, in most cases, much smaller than in the original studies. So for positive effects, the replication median size was median effect size was 85% smaller than in the original study. And for 92% of replications, the effects were smaller than the original. And then they also had these kind of binary methods of assessing whether or not the effect replicated, as well as the effect size measures. So for positive effects, 40% of replications succeeded in 3 or more methods. But for null effects, 80% of replications succeeded in 3 or more methods. So you can see that null effects were kind of easier to replicate in a way than significant original results. And overall, there was a 46% success rate, which is where that number came from in my original kind of slide about the different fields in science. So what can you do to kind of give yourself the best chance of having replicable results in your own research? It's really important to disclose how your data were collected and analyzed. So one way of doing this is the 21-word solution proposed by Simmons et al. So this is where you just include in your papers, we report how we determined our sample size, all data exclusions, if any, all manipulations and all measures in the study. So including the statement, we would hope that you would always do all these things anyway, but kind of it's a reminder to do these things and also to make it explicit that you have done these things so that people aren't left kind of guessing. Another thing you can do is consider pre-registration and registered reports. So these are ways of registering your methodology and hypotheses before collecting your data. There's lots of resources out there on how to do this, but I'm not going to be able to get into the kind of logistics of how in this talk, but there's lots of resources out there. And very important to engage in research methodology and statistics training. So this is to ensure that your studies are well powered, which I briefly mentioned before. So you don't want a situation where you get a result, like let's say you get a null result in your own work, but you're not really sure what it means because you're not sure whether maybe you just weren't powered well enough to find the true effect. So you just kind of making sure that you go into a study with the best chance to find some truth is very helpful. Okay, so moving on from replicability to reproducibility. So reproducibility, sometimes called computational reproducibility or analytic reproducibility, is the ability to recreate the same results as the original study using the same input data, computational methods, and conditions of analysis. So going back to our kind of, if we remember our table, it's same data, same analysis. And ideally, computational reproducibility should be achievable by another researcher. So using only a set of files and written instructions. So that's kind of a good test of whether something is actually reproducible if it doesn't take kind of the knowledge that you don't even realize that you have to conduct your analyses. So going back to RPCB, the kind of secret history of this, not so secret because it's in a paper, but the kind of thing that wasn't mentioned in the previous paper is that originally they had actually aimed to repeat 193 experiments from 53 impact papers, but they ended up only being able to conduct 50 experiments from 23 papers due to barriers and challenges that they encountered. And some of these are related to replicability, but a lot of them are also related to reproducibility. So I'm just going to kind of talk about them together. So the major barriers that were encountered were a lack of reported key statistics and or shared data in the original papers, difficulty in obtaining data from authors, insufficient experiment details in the original papers necessitating clarifications from authors. The majority of peer-reviewed protocols required modifications, but only the minority of these were actually implementable. And so we can kind of look at that in a bit more detail here. So they designed the 193 experiments that they originally intended to replicate. And we can see here, large majority of the data was not shared. There was some, either the analysis was not reported or the test was unknown in the vast majority of cases. The code was not shared in the majority of cases. I was glad to see that the reagents were offered in the majority of cases where this was relevant, at least. But protocol clarifications were needed in almost all of them. And it was very disappointing to see that the authors just straight up did not help quite a lot of the time and only provided kind of minimal or moderate help some of the time as well. And so then if we kind of go beyond this to the ones that were the experiments that got past this stage and were actually initiated, again, the reagents were shared in the majority of cases, but there were at least a few modifications needed, if not a lot more in the vast majority of cases. But these were not always able to be implemented. So what can you do to ensure your research is most likely to be reproducible? A big thing is to improve your documentation and reporting. So one way to do this is to follow minimum reporting standards. So there are often reporting standards for reporting different types of research. So in psychology, well, not just psychology, but you might have a reporting standard for meta-analyses or for systematic reviews or for clinical trials. So you can find a relevant reporting standard to the type of research that you're doing and follow this rather than just kind of writing your paper the way that your supervisor's written it or the way that papers you've read write it, kind of having a more formal checklist to go through of different things that need to be reported for this type of research. And really importantly, people often are like, oh, but what am I meant to do? The word limit for this journal is this, and I can't fit all that stuff in. That's absolutely fine. I don't believe that it would even make sense for the paper to include the level of detail that is necessary for conducting a replication on a piece of work. But I do think that that information should be always available. So this can be as supplementary materials. So your paper can kind of be seen as like almost a signposting to materials outside of the paper that actually form like the holistic, everything that was involved in conducting the study. And so always have this somewhere, even if it's not in the paper. Really important to share your data code and materials. We saw here that this was done very rarely in the RPCB original papers. So you can share these things in either domain-specific repositories, that might be ones that you kind of know of, that people in your field use, or generalist repositories. I've listed a few examples here, but just important above all to kind of share whatever you are allowed to share. And I found out through doing research for this talk, just because I'm so out of the loop with this because of being from a psychology background, that repositories are also available for sharing physical materials. So I thought that was really cool and something to look into depending on the type of research that you do. It's also important to conduct your own reproducibility checks. So this is basically ensuring that your analyses rerun before submitting your paper and sharing the data and code. And so it can be even better, you can do this yourself if you don't have anyone you can ask, but it can be even better to have a kind of code buddy where you just have a deal that you run each other's code to check analyses is reproducible before either of you submit a paper. And that's just kind of an arrangement that you have. So that can be really helpful. Okay, so on to the third one of the quadrant, the robustness. So robustness is the persistence of support for a hypothesis under perturbations of the methodological or analytical pipeline. So applying different methods or analysis pipelines to examine if the same conclusion is supported under different conditions. So how much do analytical choices actually matter? In a study by Botvinnik and Nezer et al., an fMRI study involving 70 independent teams analyzing the same data set. That's what they kind of sought to find out. So they tested 9 hypotheses, and each team utilized distinct analytical workflows. It ended up that there were out of the 70, there were no teams that used the same analytical workflow. And diverse workflows led to significant variation in the results of these hypothesis tests. So even for teams whose analysis plans started out quite similar, by the end, they could have very different results. And this highlighted the impact of analytical flexibility on scientific conclusions, and was very useful in identifying factors that contribute to fMRI analysis variability. But this is likely to be similar across other fields as well. So what we have here in this graph is that the pink dots are the actual results. So the x-axis is each hypothesis, each of the 9 hypotheses that we've looked at. And the y-axis is either the actual or predicted fraction of teams reporting a significant result. So the actual is the pink, and the predictions, so they were asked to predict beforehand whether they thought that they would get a significant result for each of the hypotheses, and the predictions are the blue and green dots. So you can see, first of all, that there's a very low percentage of teams reporting a significant result for most hypotheses. The outlier is hypothesis 5, for which a lot of teams did report a significant result. But you can also see that the predictions, so the blue and green dots, are much higher than the pink dots, showing that researchers were optimistic about the significance compared to the actual results. And even though hypothesis 5 was the only one that kind of got quite a lot of teams getting a significant result, the predictions for this weren't that different from the predictions for lots of other hypotheses that didn't do very well. So that's kind of interesting as well. But one question is, should we expect this? So maybe there isn't a problem with different analyses leading to different results. We know that different statistical tests make different assumptions and test different things. And using different covariates should change the results, because that's kind of the whole point of including them, is to check whether they do. But it's important to think about the kind of how dangerous it is to have such a fragile finding, as they call it in the manuscript. So a fragile finding is a risk factor for replicability and generalizability. So if your result is only present in one very, very specific analytical kind of pathway, this could be because all the things that you are controlling for in that specific analysis are exactly correct for understanding the true effect. But it could also just be that, you know, through doing loads of analyses, this was the only one that came out significant, and that's kind of more due to chance and not due to those factors about that specific analysis actually being important. So it can amplify concerns about p-hacking and overfitting. So it's possible that the reason that this result is so fragile is because it's not real at all. So what can you do to think about robustness in your own work? So one thing is to use validated analysis workflows. So you can use existing validated pipelines where these are available. I know that these are available for quite a lot of neuroimaging analyses. I'm not sure about in other fields, but where these are available, kind of using these pipelines, people have taken a lot of time and done a lot of work to validate these. So kind of it makes sense to just use those, not reinvent the wheel, and not open yourself up to so many kind of questions of why you did it that particular way, or just makes more sense to use something that's validated. But where there aren't already validated analysis workflows that will work for your particular study, you can still test your code out on simulated or secondary data first, just to make sure that you kind of thought about the structure of the data and what types of analysis might make sense. Obviously, there's always going to be cases where once you get the real data, you need to change something about the analysis, but just documenting this in a very transparent way is the best way to deal with this. It's also really important to share your analysis workflows, so share your data and your code. And this is both so that others can validate the code that you used. And I know it sounds scary, but maybe say, I don't think that's quite right. You need to control for this, or I think it would be better to use this type of model. That seems really scary because it's like opening yourself up to criticism, but it's always better to have this earlier rather than later. And in the end, kind of, it will benefit better science if we work together to make things as kind of rigorous as we can. But also this is so that others can run their own analysis with the same data. So if you have shared your data and your code, this is, maybe you haven't conducted any robustness analyses, but at least it offers others the opportunity to conduct these analyses if you have shared your data. But even better still, you can also conduct and report multiple analyses on the same data set. So this is actually something that's really common in some fields, but less so in others. So I remember being really shocked because I'm coming from a psychology background. It's very common that we will just like do one analysis for one experiment. And rather than kind of looking at the robustness of that particular analysis, usually the types of controls that we would do is running another experiment that controls for something, or or kind of like adding a new condition. But what I hadn't seen, which when I was working across the social sciences and kind of coding results from different papers, in economics, it's extremely common to have pages and pages and pages of robustness analyses. So they'll have the original result in like the first couple of pages, but then like 40 pages will be robustness analyses. And this is just normal for that field. So yeah, I think it's something that psychology can definitely think about, and other fields should be kind of thinking about as well. So you can do this on your own, or better yet, you can have multiple people conducting analyses. It can be really useful to kind of see how other people would approach the question and the data, and kind of think outside the box of kind of how you've been thinking about it. And you can also consider multiverse analysis. So multiverse analysis is where you go into it like on purpose, mapping out every possible analytic decision that you could make that would make sense, of course. And kind of when you've got this kind of map or multiverse of options for every time that you could have made a decision in the analysis workflow, you just do all of them, which is like, sounds like a lot of work, and I'll just kind of admit that I've never done this myself, but is amazing because then you're able to see how these different analytical decisions affect the result. And then you can kind of see either where there's patterns, like, oh, every time in all the analyses when we control for this, then it changes the result in this way. So we can quite strongly kind of conclude that that makes a difference, as opposed to like maybe on one of the pathways, something really random happens, but it doesn't happen anywhere else in the multiverse. Maybe that's more of a fluke. Okay, so lastly, I'm going to talk about generalizability. So generalizability refers to how applicable a study's results are to broader groups of people, settings, or situations they study, and how the findings relate to this wider context. So generalizability refers to the applicability of a research finding to other sample settings, methods, measures. So the problem is that researchers often make unwarranted universal claims. So in papers, people don't often say, I think this finding is only applicable to the exact lab that I did this in this context, in this month, in this weather. Usually they don't do that. Instead, what they do is they make grand claims about, well, in psychology, it's about human development, you know, infants learn language in this way, like, or, you know, big statements like that, as opposed to kind of contextualizing. So, and this is a problem. And another problem is that there's often a misalignment between verbal and statistical expressions. So, and this can make it unclear which features are essential for replication, which can mean that people can, you know, maybe if you go to replicate something study and you find that you don't find the same result, they later say, oh, well, it's because you did this thing slightly different, which has happened to me. But because they haven't kind of specified this in the model originally, then that kind of, it's hard to refute, because yes, it is possible, but at the same time, you kind of are only saying this once you have the replication result, which is really fair and is kind of biased. So conceptual replications, so it's important to consider the replication and generalizability to be very related and kind of more of a spectrum. So we previously, when talking about replications, have kind of been implicitly talking about direct replications, which is where you're trying as hard as possible to make the study as similar as possible to the original. Whereas once you start intentionally or not intentionally changing things about the replication study, this goes into conceptual replication territory. And all conceptual replications are tests of generalizability. So they're essentially testing whether the original result generalizes to these new conditions in whatever you have changed. So generalizing findings to previously untested circumstances. And so it could be that a finding is a true effect. So you could maybe, if you did a direct replication, replicate the result. But it could be that this result is only comes out under very narrow conditions. And these conditions might not be sufficiently defined or understood by the original authors or in the original paper. So it's hard to kind of predict which narrow conditions might be essential for replicating the effect. Also, the theoretical explanation for why the finding occurred might be incorrect. So it could be that the original result was due to some confound that wasn't present in the replication study, but kind of that wasn't the original thing that was being studied was never what was driving the original effect. Or it could just be that the true effect is much more limited than the initial discovery. Which is really important to know for further theory development and applications. So it's really important to test generalizability in order to know the boundary conditions of an effect. So how do we approach this in psychology? So I'm not sure how many of these would be relevant to kind of more biomedical context, but I just thought I'd kind of, you never know how things might be inspiring or make you think about it in a different way. So in psychology, we have several ways of studying generalizability, and so some of them are outlined here. So one is systematic reviews. So we look at the heterogeneity of an effect across sample settings, measures, methods. So in the studies that would be included in a systematic review, they're likely to vary on these factors. So if we can quantify this and work out, oh, is it the case that whenever this sample is used, we get a larger effect than when this other sample is used, for example. We also do a lot of cross-cultural studies, so we can use the same or a slightly adapted paradigm in different cultures and compare the results that we get. There's been a push recently to generalize from rather than to underrepresented groups. So in a lot of the kind of replication studies that I have talked about in the different fields, often what's happening is you're getting high impact papers that have been published in the field, and then you're checking whether those results replicate. Either in the same country or countries all over the world, but the original results themselves are most often from the global north. So many labs, Africa is an example of doing this kind of in a flipped direction. So looking at if we get studies that have been originally published by African researchers in Africa, how can we look at their replicability across the world rather than the other way around? Another thing that we can do is big team science. So this is a way of getting labs across the world to conduct the same study and pool their results together, which can be really great, both for just getting a really large sample size, but also for looking again at the heterogeneity of an effect across the ways in which these labs may be slightly different from each other. And we can also look at predicting generalizability. So we only talked a very little bit about predicting results when the researchers were being optimistic about when we were talking about robustness. But we can use prediction to basically investigate whether researchers can predict which effects will and won't generalize in the same way. And so this can be really useful if we find out that researchers are very good at this. They could maybe be used as like a first pass of working out where maybe more research is needed for replications and generalizability studies, and where maybe researchers feel that this isn't needed because the result is quite strong. So it's kind of useful to know how accurate researchers can be at that. So if you're interested more in generalizability, you can listen to the report on our workshop, which is linked in the slides. So what can you do? One thing you can do is include constraints on generality statements. So this is to basically identify and justify the target populations for the reported findings. So in your papers, you basically say, we think these results would generalize to these contexts. We're not sure about these other contexts. And it's not that you know that this is true, but it's important to kind of have the researcher contextualize the results at the point of actually having that paper out there, as opposed to what I talked about previously of like having that as a post hoc thing of like, oh, well, you can replicate our results because of this. You should kind of have that thinking before you see the results of any potential replication. It's almost like when you're writing your papers, you should be imagining if someone were to do a replication of my work, which context would I predict they would find the same result and which context wouldn't I? And that's kind of a thought experiment that you should be doing anyway before it actually happens. You should also have open materials and a detailed description of methods, so that anyone trying to replicate your work will know exactly what you used, and that any deviation would be on purpose. So if they're doing a conceptual replication, they would know they're doing a conceptual replication, as opposed to just it being conceptual because they didn't know exactly how to make it direct. And then there's a couple of methods you can use to actually actively embrace generalizability. Again, not sure how applicable these would be across different types of research, but just in case they're useful to you. So one is radical randomization. This is kind of mapping out all the experimental variables that might moderate an effect and just randomizing them and having kind of this very noisy data set on purpose, so that if the effect kind of comes through all of this variation, then it must be robust to this to all the kind of features that you have manipulated, generalizable to all those different contexts. And then integrative experiment design is where you map the design space of possible experiments and iteratively test theories. So having a more programmatic research approach as opposed to individual studies, kind of thinking about how the results of several studies put together would answer your question. So just to conclude, we've talked a lot about different things you can do, and I think they can be kind of summarized into a few categories. So most important, be transparent. So this is in regards to your writing. So disclosing how your data were collected and analyzed, following minimum reporting standards where these are available, including constraints on generality statements. And then also be transparent in terms of what you're sharing. So as much as possible, share your data, code, and materials. Conduct reproducibility checks on these materials. Register your methodology and hypotheses before collecting data. And then, as well as being transparent, you should also work really hard to improve your statistical methods. Ensure your studies are well powered. Use validated analysis workflows where these are available. Conduct and report multiple analyses on the same data set. And consider research methods that embrace generalizability. So thank you for listening, and thank you to many of my collaborators whose work I presented today. And yeah, happy to take your questions.